קליק קפדן בזמני? איך הוא, למה? מה הוא אומר שאם לא תהיו בזמן אני לא נותן לכם להיכנס? טוב. So, we are few but high quality. What? טוב. Okay. So let's see. Can we dim the lights a little bit more? Is this okay? Right. It's cold around here. So good morning. This is eventually the second and last time we meet formally. And what we did on Tuesday, I guess, right, was a kind of mega introduction to many of the basic processes in the development of the nervous system. And I decided to give you today a little bit more about the peripheral nervous system, about which we just talked a little bit. I told you that the neural tube is the progenitor structure for both the PNS and the CNS. However, the PNS develops in an entirely different manner. And this is what we are going to do today. I'm going to divide the talk into like 3 parts. A very, very short part, which is going to be introductory. A second part, which is going to deal a little bit with evolution, equals a little bit of hand waving, which is nice. And the third part, which is going to be rather brief, in which I'll tell you a little bit of our own data and how we do approach the development of the PNS. So let's go and see some things that I guess you should already know. This is the process of neurulation, namely the process by which the ectoderm transforms into a neuroectoderm. It begins folding in the midline, in the dorsal midline of the embryonic body. Then the neural folds, which are these ones, begin approaching each other. And we know, obviously, that after approaching, the last stage in the formation of the neural tube is actually the fusion in the dorsal midline of the 2 folds of neuroectoderm, the regeneration of the epidermis on top or dorsal to the neural tube, and basically the separation of the primordium of the nervous system from the epidermis. Now, a structure which will become important in a few minutes, which I would like to remind you, is the notochord, this rod of cells which spans most, but certainly not, the entire length of the embryo. Now, let us go to the peripheral nervous system, because if we perform a cross-section, okay, we cut our baguette, okay, into slices, right? Transverse like this. So it's like slicing the hodu. And what we see is the neural tube, a cross-section, the notochord, and this is the mesoderm on both sides, which will eventually become muscle, vertebra, dermis, and so on and so forth. And it appears that at the dorsalmost portion of the closing neural folds, this area, which is colored here in dark blue, there is a specialized population of cells that we term neural crest, because it's the crest of the area, neural crest. And these cells are going to be the primordial cells of most, but not all. Okay, I would like to stress that, even if we don't have time to discuss that, of most, but not all, of the peripheral nervous system. Now, why are these cells dark blue and not light blue? Because already at this very early stage of closure of the neural epithelium, these cells are molecularly different from the rest of the neuroepithelium that will become the central nervous system. What you're seeing here is what we call an in situ hybridization. Do you know what that is? I'll explain that for the sake of those who do not know. It's a kind of reaction, RNA-RNA, by which we label a certain RNA, we prepare a certain plasmid, a vector, and we just hybridize it. We put it on top of sections, okay, or of whole embryos. The RNA is obviously an copy of the normal RNA produced by the cell, and we then develop this by virtue of a certain biochemical reaction. So it becomes blue, it may become other colors depending on how we develop it, and we basically see the expression of different species of RNA molecules. So this is an RNA encoding for a gene which is called SNAIL2, and there are many additional genes encoding for different receptors and mainly for transcription factors, which are already expressed there. And this means what? What I said before, that this dorsal-most region is already distinct, at least in certain terms, from the rest of the neuroepithelium that is fated to become the central nervous system. Now, what differs from these cells, between these cells and CNS cells? Although CNS cells are going to migrate after exiting the cell cycle, these cells are going to behave completely different. They are going to undergo a process that we mentioned also on Tuesday, which is a process called EMT, epithelial to mesenchymal transition, by which epithelial rigid cells will become motile. And when becoming motile, the difference between CNS and PNS progenitors is that crest cells will leave the area where they were born, and they will engage in a very, very extensive migratory process that will lead them sometimes very far from the region of origin. So they will become basically mesenchymal cells, pretty much like our muscle progenitors, our cartilage progenitors, and stuff like that. So they will intermingle among cells in your body before they aggregate into ganglia and so on and so forth. We shall reach this derivative issue in a second. So what you're seeing here is a process, and this is a cross-section through the brain, okay? In this case, through the midbrain, you see that the neural tube is flattened. The central canal is much larger because this is going to be one of the ventricles. And look at this. This is CNS. This is a nuclear stain, by the way. And look at these cells here. These cells have left the neural primordium. This is what characterizes the delamination or emigration of crest cells in the head. It's an unmasked type of behavior, contrary to what happens in the trunk, where in the trunk, cell by cell, do live independently the neural tube. They arrange in like chains. They contact each other by filopodia, pseudopodia, lamellipodia, so they are not isolated. By far, the process of emigration is an isolated process, whereas in the head, it's an unmasked dissociation of cells from the neural primordium. Now, you can see here the onset of emigration in another region of the body, in which you see like two tears leaving the neural primordium. So these cells are very particular, and they begin as a very small and discrete population in the dorsal neural tube. However, they diversify into a huge amount and huge variety of derivatives. And this is why this issue, this theme of neural crest, has become, during the past 20 years or so, one of the most attractive themes to understand developmental biology, namely to understand how a seemingly homogeneous population of neural progenitors diversifies into so many types of derivatives. And this is a very basic question in development. So you can argue that certain cells will give rise, for example, to, I don't know, cardiomyocytes and cardiac jelly. But these cells give rise to all these list of derivatives, and I'm going to mention it very quickly, but it's important that you somehow, I don't know if remember it, but pay attention to it because it's very interesting and relevant. And then I'll tell you and I'll teach you how do we know that these are the derivatives? How do we follow the fate of the cells? How do we map them? Okay? But first, the bottom line. So the peripheral nervous system is almost entirely, but not completely, derived from the neural crest. And as I guess, have you learned already PNS? Do you know anything about the PNS? Yes, so it's going to be easy. Don't hesitate to ask if anything isn't clear. So I'm going to do it quickly. Okay? So all the spinal ganglia that are located on both sides of the spinal cord, neurons as well as glial cells derive from the crest. These are the dorsal root ganglia. Then the entire cohort of cranial sensory ganglia, trigeminal, jugular, nodose, you name it, facial ganglion, vestibuloacoustic, all those ganglia are derived from the crest, but in a complicated way. Not all the cells are deriving there from the crest. All the glia is crest, but the neurons have a mixed origin, and this is how I restricted my statement before. The neurons may come either from the crest or from an additional structure called the ectodermal placodes. Let me just draw it for you. If we have here a section through the brain, because we are talking only about cranial regions, what we see is that the crest, okay, will organize, for example, let's look at the trigeminal ganglion, which is the most complicated one. The trigeminal, you know, has an ocular component and a mandibular component. So the ganglion sends fibers both to the eye as well as to the upper jaw. And this is the ganglion. We know that all the glia come from the crest. This is known, glia from crest. But the neurons, there are several types of neurons. There are proprioceptive neurons, as in any sensory ganglion. You know what proprioception is, okay? There are nociceptive neurons transmitting pain. There are mechanoceptive neurons of all kinds, pressure, etc., etc. So these different types of neurons come from two different origins. It appears that the nociceptive neurons come from the neural crest, but the proprioceptive and the mechanoceptive come from the ectodermal placodes. Now, what is a placode? This brings us back in evolution to older animals. The placodes are ectodermal thickenings. So if you take the ectoderm in this region, which you would argue generates only epidermis, it appears that in the head we have several placodes like that. It's a thickening of the ectoderm, pretty much like this which generated the neural tube, but laterally, on the sides, and not in the midline. And this area produces cells which delaminate and enter to contribute to the formation of this ganglion. So this ganglion is contributed, if I am going to an earlier stage, by cells coming from the crest and by cells coming from the placode. So it was mapped by means that you will understand in a few minutes, that different types of neurons, which were then mapped physiologically and molecularly by virtue of their expression of different TRK receptors, TRAC receptors, neurotrophin receptors, NGF, BDNF, NT3 receptors, TRAC-A, TRAC-B, TRAC-C. If you heard about them, if not, never mind. I'm not going to complicate it any further. It has been found that this is the derivation of those cells, and that it has also a very important evolutionary significance, by which it appears that the primitive cells were placodally derived, and the crest cells, which are the nociceptors, are more modern in evolution. They appeared only in bony fishes, but in cartilaginous fishes, basically their ganglia, their sensory ganglia in the head, do not contain nociceptors. it's very interesting. So it appears as if the sensation of pain would be a kind of more modern sensation that evolved pretty late in development, but as usual, I take evolution, as I said, as a little bit of hand waving. We have things that we can really think about them and argue half philosophically, but this is the current idea about them. So as I said, all the satellite cells, which we call glia, okay, do come from the neural crest. This is without any exclusion. And there are now many, many other additional things that are being found. I'm not going to develop them today. The entire autonomic system develops from the crest. The autonomic system means all the sympathetic ganglia of your body, all the parasympathetic ganglia of your body, neurons and glia comprised, and a very important nervous system, which I teach a lot to medical students because it's an amazing system that we used to call the second brain, and this is the enteric nervous system. The gut develops most of it from the endoderm, but the musculature of the gut comes from the mesoderm, and the entire innervation of the gut comes from the neural crest. And this comprises two things. It comprises an inner network of plexi, of nerve cells, glia, as well as peptidergic neurons and all kinds of stuff, many, 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 many types of neurotransmitters. We have a number of neurons in the enteric nervous system similar to that in the spinal cord. So it's a second brain by all means. And then it is composed not only of this intrinsic nervous system, but also it has a control by which systems? What does control that our bowel moves faster or slower? The peristalsis. Which system controls the peristalsis of the bowel? If you are nervous, if you have an exam and you run to the toilet, which is the system that controls that? What? No. It's much lower. It's the sympathetic and the parasympathetic systems, which are also derived from the crest. So you have a sympathetic and parasympathetic control of the gut. And there are a lot of diseases which are very, very specific to the gut, which are very specific to populations, for example, of Ashkenazi Jews, like Hirschsprung's disease, in which crest cells fail to migrate to the large bowel, to the large intestine, and this causes what we call an aneural megacolon. Namely, these kids are born with the intestines which are okay, but the distal part of the intestine, which goes to the rectum and the anal portion, is distended this way. It may be like a balloon. Why? Because there is no tension, there is no contractions, there is no nervous system there. So these kids have to be operated, etc., etc., and depending on the gravity of this thing, the approach is obviously different, and also survival is also different. But this is a very, 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 very crucial and important part of the nervous system, which, by the way, in many departments of neurobiology, people neglect. But there are so many researchers working on that, and it is so challenging intellectually that you can't imagine. It looks like, dealing with the intestine, but it's very, very interesting. So this is just to tell you that when now you run to your toilet, think crest, okay? Now, the parasympathetic and sympathetic innervation not only deal with your toilet stuff, they also deal with your heart, because before an exam, your heart rate is increasing, and you are sweating a lot because all your sympathetic and parasympathetic systems also innervate your sweat glands. Okay, so these are very, very important functions that are performed by a system which is autonomic, which you do not control. You wish you would, but you don't. Schwann cells. We said last time that glia is central and peripheral. Peripheral glia is Schwann cells. They are all derived from the neural crest, and they are those cells which myelinate the peripheral nerves. There are now a lot of ongoing studies on Schwann cells because it has been found, and this is something amazing, it has been found, and we have also been associated to that, that Schwann cell progenitors, not differentiated Schwann cells, but Schwann cell progenitors are also able to generate pigment, which go to the limbs and the abdomen, whereas pigment in the dorsal part of the body directly derives from the crest, and also neurons. It appears that a certain type of parasympathetic neurons in your head comes from Schwann cells, which is amazing. This is completely new. It appeared now back to back, and I wrote an article on that. You can see it in Science by the group of Adameiko, a group from the Karolinska Institute in Sweden. So Schwann cells derive from the neural crest, and then there are quite a lot of endocrine and paraendocrine derivatives. The most famous, like some thyroid components and parathyroid, calcitonin-producing cells, and so on, but the most famous ones are the adrenomedullary cells. We have here, on top of our kidneys, two glands, which are called adrenal glands, right? They have a cortex, they have a medulla. Medulla is the center, the heart. And the medulla produces chromatin cells, and they derive from the neural crest, and this basically mediate the fight and flight response of our bodies via secretion of different hormones. So I'm not going to talk about that anymore, but as I said, pigment cells. This is something which is extremely important. I mean, we don't know why. We don't know why. A structure which otherwise generates neural derivatives, why does it generate pigment? And eventually, this is also of evolutionarily significance. I'll tell you a little bit about our studies, which try to understand how a crest cell decides whether to become a neural cell or a pigment cell, but if we have time, we will reach that by the end of the talk. And finally, I would like to talk about all this, you know, in one word. This is called mesectoderm. This is called mesectoderm. What is mesectoderm? Ectoderm we know, right? Ectoderm generates nervous system. But mesoderm generates muscle and bone. So what is mesectoderm? It's a kind of chimera. And it appears that the neural crest, only at head levels, is able to generate the bones of your skull. So these bones here, okay, up to the basiocciput, they all come from the crest. So why? 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 You asked why.
I never asked why.
You won't ask again why. And I'll tell you. Okay, never mind. I'm joking. So there is a model. Again, this is evolution. This is the only evolutionary part I'm going to touch upon. And this model says the following. It's called, let me go a step further. It's called The Neural Crest and the Origin of Vertebrates. It's a beautiful paper that was put forward by two scientists, Carl Ganz and Glenn Northcutt, an old paper, which is still relevant, which is still very, very relevant. And it claims basically the following. I'm going to go back to the picture I showed you before in a minute. It claims the following. The notochord, which is the structure I asked you to remember, runs along the entire body. I taught you that the notochord is responsible for inducing the ventrodorsal patterning of the neural tube, but as I said, sonic hedgehog coming from the notochord is like a sun. It irradiates and it is secreted all over. So it affects not only the nervous system, which is up, but also the mesoderm, which is on both sides. And upon signaling of sonic hedgehog, the mesoderm, along the entire body, will generate bone. Now, the notochord does not reach the entire brain. It ends up at the area between, which approximately goes to the diencephalon, around here in the embryo. So all this area of the brain does not contain notochord. But so you would argue that the mesoderm of the head does not receive a trigger by sonic to produce bone. So mesoderm in this area would be unable to produce bone. Now, let us leave this for a minute. During evolution, there has been a very important process which is called cephalization. You have seen that in the first slide I showed you. Namely, the size of the head, okay, has increased enormously up to ours and up to that of primates. So this process of cephalization, which means the development of a head which is bigger and which concentrates most of our senses, vision, audition, taste, etc., needed a kind, this is the hypothesis, needed a kind of protection by virtue of an osseous box, something that would protect the growing brain. And in evolution, we know about cells which undertake a different function based on the Darwinistic type of need to survive. So the theory says that the neural crest took over the function of the mesoderm, and even being an ectodermal structure, it generates mesodermal derivatives. And these are bones and dermis, not muscle. Bones and dermis of the head and the neck region are generated by neural crest. So what they say is this is in the transition between protochorders and vertebrates. It's the acquisition of skeletal tissues. It's the development of the brain, which needed a protecting head on top of it or around it with sense organs, and also a jaw as an organ of predation, because your jaws are also neural crest. Because the idea of this new head hypothesis says that this happened exactly during the transition in which animals became predators. And when you become a predator and you need to eat another animal, what do you need? You need jaws and you need teeth. So part of our teeth are neural crest as well. So the neural crest in the area of the head, but not in the trunk, will generate the skull or parts of the skull, not all of it. It will generate part of your teeth. I am not going to detail on what. It will also generate your jaws. Okay? So let us see this in a little picture. This has been mapped precisely in chick embryos. Okay, and what you see here, this is a chick embryo, this is the beak. In a minute. This is the beak. The beak in the chick also comes from the crest. Okay, so this is the calvaria bones and all these bones, except for the basiocciput, which are these bones here, which come from the mesoderm. Okay, so you have here a full mapping of those derivatives, and by analogy, in the human being, the lower jaw, the upper jaw, the orbit, and all these bones of the cranium, except the basiocciput and this other spheno whatever, which I don't remember, are coming from mesoderm or other type of derivatives. So this is the idea behind that, and this explains why the neural crest, how the neural crest, I'm afraid to use this word here, how the neural crest is able basically to generate derivatives which are not neural exclusively. Ken, Amir.
I have two questions regarding the previous slide. You said...
This one?
No.
Ken.
You said that the trigeminal nerve, there's this...
Trigeminal ganglion.
What?
Not nerve, ganglion.
The ganglion.
Of course.
Okay. There is this differentiation between the sensory receptors, and the nociceptor receptors, and the...
proprioceptive, for example.
Yeah. But is it true just for this ganglion, or all over the body?
Okay. No. As I said, the DRGs, which also have proprioceptive, nociceptive, everything comes from the crest.
Oh.
But this is a good question. Let me say that if you look at an embryo, evolutionarily, the embryo can be divided into three. The body, which is the most primitive area, the neck, which is an intermediate area, and the head, which is a new phenomenon in evolution. So it is interesting that even in the trunk, I told you that the neural tube, if you ask, you're going to get an answer. If the neural tube closes like that, and I told you this is a sheker lavan, this is half of the truth. Why? Because in human beings and in higher vertebrates, the central nervous system is generated like that only from the head till your umbilicus. But all the areas of the spinal cord that go from the lower thoracic area to the lumbosacral area are generated like in fish by a mechanism which is completely different, and it is called cavitation. So what you have is a kind of ectodermal mass, which then undergoes epithelialization, a hole in the middle, and then you have a tube. No process whatsoever of folding. So the lowest part of your body, not the lowest, the caudalmost part of the body, even in human beings, resembles the fish, in which the neural tube is not called neural tube, it's called neural keel. It's different. But then we begin resembling more and more advanced phenomena or advanced processes. So even in our own body, okay, from the tail to the head, we can evolutionarily distinguish different things. I said that you can divide it into different parts. Because of considerations I don't have time today to make, in terms of the behavior of neural crystals, we know that they are different. And when you look at the head, the behavior of crystals is totally different from that in the trunk. And this is a very, very interesting issue in terms of how they delaminate and how they proliferate and how they are fate restricted and so on and so forth. Now, so DRGs, everything comes from the neural crest. Neurons of all kinds and glia. Now, in the ganglia in the head, the trigeminal is the most complicated because neurons are of mixed origin, glia are from the crest. But if you take another ganglion, a sensory one, for example, the nodos or the jugular, the neurons are all placoidal, and the glia is all crest. So you don't have a crest. So every ganglion is unique in its composition. I gave the trigeminal because it's the nicest example of the most complicated type of contribution. But every ganglion, you should know yourself where it comes from and which contribution. Of course, I'm not going to ask you that. It's only a matter of principle. Beseder? Okay.
So it's kind of a question of why, so I have to be careful.
No. Ask why. I'm joking. Really, I'm joking.
So we saw that the bones in the skull are from a different origin than the other bones. So they come
Right.
from the crest and not the mesoderm.
Right.
And what that paper said is that it's a later part of evolution, but is there any reason to think that, okay, so I need a new bone to protect the developing brain, but why shouldn't it come also from the mesoderm one? Is there any...
Well, yes, the explanation that they provide, again, hand waving, is that there is no notochord in the head. That the notochord, which is the inducer of bone, does not exist in the head. This is the explanation they provide, right? Because the notochord via sonic, I said that before, maybe you missed it, the notochord via sonic, for example, if you have here, okay, let's say this is the tube in another area, you have the notochord. So the notochord not only influences the tube, it influences the endoderm, but sonic also goes to the mesoderm here, okay, around the ganglia, and it produces sclerotom, which is the primordium of the bone. So if you don't have a notochord here, obviously you will not have those, and the nervous system develops differently in the head than in the spinal cord, right? Okay. Sonic is there, but not in the notochord. Sonic is within the nervous tissue exclusively, so it acts locally and not on the adjacent structures. Okay.
How exactly can we trace back the origin of all these cells?
This way. Now, there is a French scientist that I had the honor to be in a postdoc in her lab, and what she found is the following. She says, how can we trace, how can we give you this table? I mean, this table did not exist till she didn't do all these studies. And this is very funny, so I want to share it with you. It's a classical work. And I think it's very interesting. Now we have many other ways. But before we had specific markers for tracing cells. And again, a marker, a molecular marker, is a jumping epitope. It is never expressed only in a single type of cell, usually in more than a single type of cell. So usually, I mean, you cannot say that this is a lineage derivation necessarily, so you have to be very careful. But in order to trace back the development of a cell to its progenitors, you need what is called a stable marker. A marker which is stable, a marker which does not disappear by dilution, and a marker which does not harm normal development. Okay? So she noticed, by working with embryos, she worked, it's a very interesting story, she worked in Paris, near Paris, in Nogent-sur-Marne, in the lab of a very famous Jewish teratologist who dealt with monsters, basically human monsters and chick monsters. All this, we have it a lot in the lab, all kinds of malformations. So this is a field called teratology, all kinds of diseases which called malformations of the newborns. And she was his best student, and then she inherited his place. And it was interesting because it was a time in Paris, in France, in which women would not inherit any position. So she married a guy who was a cardiologist and an expert in cardiology, in research in cardiology, and basically he got the position. Not she, although everybody told me that she was much better than he, but he got the position, and as his wife, she got a position in his lab. But later on, it took her, you know, a lot of strength, and this is my only feministic approach to you, a lot of strength and a lot of fighting till she divorced this guy and basically took over this institute. It wasn't a lab, it was an institute of hundreds of people. And I mean, she's fantastic. She's one of the, most accomplished scientists I ever knew. So her name is Le Douarin. This is still, she keeps the name of her ex-husband. And what she observed is the following. She was dealing with chick and quail embryos. So she observed that the nucleus of the quail embryo is different from the nucleus of a chick embryo. They are completely different. I'll show you exactly how. These are nuclei of the quail, and these are nuclei of the chick. You can readily distinguish them. The nucleus of the quail has a kind of heterochromatin slab. Heterochromatin which is very condensed. And what you see is a circle, a little ball. And the heterochromatin of the nucleus of the chick is very dispersed. So if you stain them with a certain nuclear stain, and now we have a pretty antibody, which is called QCPN, and QCPN will only recognize cells from the quail. So now, if you go and you perform a graft, let's do it this way. You perform a graft by which you go to a very early embryo of the chick. You remove a portion of its neural tube before the crest has left, namely that still contains the crest. And you place instead the neural tube with the prospective crest of a quail. You close the embryo, and this is what we do in the lab. We close the embryo with sellotape, masking tape. You put it back into the incubator at 39 degrees, and the embryo continues differentiating. And it even hatches, as you will see in a minute. But all the neural crest derivatives will have now, in this particular area, the quail marker. So we are able, by virtue of sectioning serial sections of the embryo, to analyze and to see exactly what is the contribution of the neural crest, the only cells that leave the CNS to go to the periphery, which are the contributions by the neural crest. And if you look, for example, I'll show you here, this is what you saw before, and this is QCPN, this antibody, which is much more effective. And this is what? What have we replaced here? This is quail. It's not the crest. This is a somite. You remember these balls which are on both sides of the tube? This is a somite. This gives rise to the cartilage, and this gives rise to the muscles. So this is now quail. Look how easy it is. So this is already a section. We can grow these embryos for as much as we want. They will develop a kind of neurological graft versus host disease only 15 days after hatching, so we have plenty of time to see what are the derivatives. And this is the way in which this table that I showed you was built. And this is what happens with the chimeras. So this is a quail. This is a quail. It is the japonica quail, the Japanese type of quail. It's brownish. It's a very aggressive type of bird, by the way, very aggressive. When my kids were small, I brought them into my house in Paris. So I brought the eggs, and we hatched them. So we had a few quails, and we rented a very big house where I think it had 100 years, where I think the owners had a lul. They grew pigeons and chickens in the garden. So we put them there, and they ended up eating each other. I mean, it was amazing. Whereas the chickens were okay, the quails ended up, and it was the female which ended up in eating the neck of the male. I don't know why, but they are quite aggressive type of aves. So these are the chimeras, and this is the white leghorn chick that serves as a host. So we can do chick into quail into chick, okay, but it's much easier to see quail into chick than the opposite. So what do these chimeras tell us? Where did we graft here? Now that you know the derivatives. Yeah, it's not only the wings, it's the entire trunk is coming from the neural crest because it keeps the pigment of the quail. The white leghorn is not pigmented per definition, okay? So what you can see is that the chimera is a chimera, and basically you can either section these ones or section them when they are still embryos and see exactly how we build this famous table. So this was one way. Now we had additional ways to do it. What we know to do in the lab, but this has already been done by other people, but we do it now by misexpressing genes. We know how to misexpress a GFP encoding vector, okay, just to trace normally the cell, to very small groups of cells, to large groups of cells, and we know to do it clonally. We know now how to manage to deal with single cells, okay, and trace the fate of single cells, as you will see in a minute. Okay? Should we? Up to them? A break? Okay, so let's make a break and then we'll continue. כן. 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 אכן, אכן. יש מידור. יש מידור. אה.
באותו כבד אתם לא צריכים לשים את זה במערכת אחרת כדי לראות לאן זה מתפתח. אתם לוקחים מקוול ואתם שמים את זה בצ'יפר, נכון? אבל אם הייתם לוקחים מארקר, פשוט הייתם מחזירים את המארקר? המארקר הוא טבעי לקוול. הוא טבעי לקוול, אוקיי. זה חלק ממנו. הבנתי, אוקיי. זה חלק ממנו.
זה מה שנגיד,
כן,
חלב.
זה
למדה כתוב
כאילו
כן,
עץ.
זה
January, end of January, there's only 1 week to that.
כן, כן.
תשמע, היא הייתה דוברת צה"ל, אני לא יכול
יש דבר,
קודם כל המון מדויקים.
Yeah, last semester, January 26th.
עבור כל X, אני חוסמת על כל ה-X, ומה שהייתי בוחרת בהינתן ה-X הזה, אבל בפועל כאילו מה שהייתי בוחרת בהינתן ה-X הזה, אם באמת ה-X הזה מספיק. פשוט באמת עשיתי, אז basically
There are people who don't want to recommend themselves. Because they feel like too much. Yeah.
הלמדות זה רק,
זהו.
שאל אותה רק שני מקרים, זה לא
So they have like a program.
That's not why I don't want to send these commercials.
כן, לא דילגתי פה.
First of all,
וואי,
בסדר, זה
Cousins of myself in the middle of a class and I'm like, I don't remember what I'm watching. It's horrible. Now,
אז רק, כאילו, מה היית בוחר? לא, דלתה, שתקרא את המדיניות של X, או X, מה היית בוחר? בהינתן שבאמת ראית X.
זה לא זה?
תסתכל על
לא מבינה. עבודה.
הבחירה לפי X בהינתן X.
אני רוצה אבל להפוך את הארץ.
חושבים שעניתי להם, אבל תנו לי דקה לענות.
אני רוצה להגיד משהו, שאתה
So, in the last hour, I would like to deal with a very important developmental question, which I hope you'll enjoy, and is a question which is very well adapted, I think, also to modeling approaches by people who have the background that you have. And this is the question of multipotency versus cell determination. One of the basic questions, again in development, of all the systems is whether a particular progenitor at any time is committed to become a certain derivative, or whether all the way, and I'm presenting you the two extreme possibilities, whether all the way until colonizing its organ or its target organ, is it a multipotent cell that is able to be changed by the environment. So this is the very extreme question, and let us apply it to the neural crest. Are crest cells multipotent progenitors, or are crest cells fully specified progenitors in terms of their fate? Or is the neural crest a mixture of pluripotent and specified progenitors? And basically, what is the dynamics of its behavior? We were talking just with Mickey about your background and the suitability of your background to deal with questions other than, higher brain functions and things like that. And I told you also Tuesday that many people are entering the modeling issues in developmental neurobiology, and this becomes a very hot and a very popular field. So these are one of the possibilities to try and really, and you'll see it in a minute, to understand how many progenitors do we deal with, how can we model the dynamics of cell delamination vis-a-vis fate determination, and stuff like that. These are very, very interesting and open questions. So there have been several approaches to tackle this idea and these questions, and I'll make it rather quick, okay? A population versus clonal approach, a fate versus developmental potential. What do we mean? If I'm going to tackle the potential of a population, what people do usually is they change the place in which a cell is born and ask whether in an ectopic place will it develop into the same derivative, will it recognize the cues to migrate to a different place and then develop as it should have developed, or will it respond now to the new cues that are given by the new environment? And to this end, the classical experiments were performed by the Duran lab, in which, for example, pieces of tube of the trunk area were grafted into the head or vice versa. And then they asked, will the crest cells develop according to the new program or according to the old ones? And basically what they got is an idea that was promoted and reigned in the field for many years. And they said, if I graft a piece of trunk in the head, for example, except for mesectoderm, which they don't give rise to cartilage and bones, they can give rise to sensory ganglia in the head. Now we know that trunk crest is able to generate glia, so it's not surprising that if we graft it into the head, they will generate glia. It's not surprising either that they will generate different types of neurons. Does this mean that they are multipotent? Not necessarily. They also observed death of cells, but they didn't know how to interpret it. Is it because of the graft, or maybe is it because some of the cells were highly specified, and when they didn't find their proper cues to develop, because instead of being in the trunk, they were in the head, they died. So all these population experiments, what we call population approach, did result in a kind of misinterpretation of reality. But this reigned in the field for 20 years. So everybody would say in the field that neural crestals are multipotent progenitors. And in many papers you can still find the neural crest is a multipotent population of progenitors. So it's true. At the level of the population, if you take the crest as a population, it's multipotent, of course, because cells will give rise to A, B, C, and D. But the question is, what is the fate of a single cell? If I go to a clonal approach, okay, not to a population approach, will I get the same outcome? Now, it's very difficult. It seems in vivo, in higher vertebrates, to perform a clonal grafting of one single cell. Why? Because it dies. Cells like to be in families, like us. So if you isolate a cell from the others and you graft it, you bring it into another side of the neural tube, it's going to die. A very skilled person called Mario Lefebvre has tried to do that for years, never succeeded. And nobody succeeded either. It's very, very difficult, not because of the technical part, but because of the conceptual idea of taking one epithelial cell and transplanting it into another epithelium. This is feasible with mesenchymal cells, not with epithelial. Anyway, so clonal approaches had to be different. And people began by asking, in a clonal approach, something in vitro, in the dish. And they said, what would happen if we isolate a single cell and we put it in a dish? Will it give rise to one derivative, two derivatives, all of the derivatives of the crest? Now, here comes the second conceptual point that I want you to understand. This is very important. What are we testing in vitro? If I take a cell and put it in a dish, and I come back 10 days later, this cell has transformed into 10,000 cells, and I ask what are the derivatives. Am I following fate, or am I following potential? And I'm going to explain what each concept means. The fate is what a cell is becoming under normal conditions. Namely, smallest possible intervention in the embryo or in whatever system, and I follow the normal fate of a cell. And developmental potential, can somebody help me?
Might be what you told us last week or last 2 days ago about the cells that you take from the, I think it was the neuromuscular cells maybe, that you can take out and then stitches it, they become many different cells, they can become these stem cells, they can become fatty cells. Does that
Absolutely.
happen or?
So the developmental potential is what a cell can become if I challenge it with an environment that is not the normal one. It's potential. What does it mean, a potential in a child? What would this child become if we give him piano lessons and English and French and Italian? Okay, it's the potential. We have a certain potential, and basically the fate of a cell is usually lower or higher than the potential. Lower, of course, because a cell will end up being a neuron of a certain type. But being a stem cell, it had the potential to become many others. Or if this cell would have been in another environment, getting another synaptic connection, it would have become maybe another type of phenotype, okay? Another type of modality. So the potential is what you can do if you are given or challenged with external conditions. So what people did to begin with at the clonal level was challenging the potential but not the fate of cells. So this is an experiment done years ago in which a certain group took one single cell from the crest and put it in vitro and gave this cell an array of factors that ensured its proliferation, its survival, hormones, growth factors, many, many things, clearly potential. And ask, what are these cells going to become 10 days later? Okay, by the way, I gave somebody here the 2 presentations, so he promised me to pass it on to all of you. So you're going to get this. You don't have to write or something, you have them. And look at the results. There is one cell 10 days later, or 0.3% of the cells, of the clones. What is the idea? You have a multi-well dish of 100 wells, okay? And you have many of those. And in each one of the dishes, you put one cell. One cell, one cell, one cell, and let's say you have 4 dishes, 400 cells. This is your 100%. They have more. Okay? You let them grow for 10 days, and then you analyze the clones. Of course, after a few hours, you come back and you see, did I really put one cell? So in those wells in which there were 2 cells, if they didn't know whether it is one cell that proliferated already or one, they threw it away. But they were left only with one cell after a few hours, and then they continued with it for 10 days. And 10 days later, it appears that in 0.3% of the wells, namely 0.3% of the clones, they found 1, 2, 3, 4, 5 type of derivatives at least that they could analyze, because they are also limited by staining, how many antibodies they can add to identify different stains. So they saw glia, neurons, melanocytes, fibroblasts, and cartilage. They took it from the head, by the way. This is why they saw cartilage. Only in 0.3% of the clones. And this is what we would call the multipotent stem cell of the crest. Why? Because a single cell was able to generate at least 5 derivatives of different kinds. Then, and this is interesting, 80% of the clones had mixed type of derivatives, namely glia and neurons, or glia melanocytes, glia and fibroblasts, and so on, or 3 cell types or 4, in stochastic combinations. And this would mean that part of the neural crest, most of it, at least in vitro, under the best conditions you can give a cell, would be intermediate progenitors, namely cells which have restricted their developmental potential to a certain extent. and are able to generate only 2 out of the 5 possibilities. But then they also got, in 20% of the clones, which is a lot, monopotent progenitors, namely only neurons or glia or melano or fibroblast or cartilage, or, exclusively. And this means that even when testing potential, even when challenging these cells with the best possible food, these clones were completely restricted to generate only one type of cell. So the conclusion of this type of experiments at that time was essentially that the crest cell is composed, or the crest as a whole, as a population, is composed of a mixed type of progenitors, some of them multipotent, and all the gradients still the monopotent situation. Now, I must say that this experiment had later on a twist. This was performed by Ledoirin and had later on a twist by somebody in Oregon, another father of the field called James Weston. And what he did is something more clever. He took crest cells, he took the tube, put it in a dish, and let the entire population of crest cells emigrate. Okay, so the crest cells become mesenchymal, they emigrate on the dish. But the point here was that they emigrate as a cohort of cells, as a group, as a cohesive group of cells. And only a day after these cells left, a few hours after these cells left, he labeled with a certain color, never mind which one of them. The difference between this and the other experiment was that the cells were not isolated enzymatically and put as a single little cell in a dish, but they were basically left while living with their neighbors. But he followed the fate of the labeled cell exclusively. And what he found is that most of the cells were restricted. And only a very, very tiny population was multipotent. So even in vitro, when doing a kind of clonal approach, but when the cells were accompanied by their peers, he found that the cells have already a certain tendency of restriction, a certain bias to become A and not B. So even in vitro, this is discussed. Let me now go and show you results that we got in the lab, which refer first to the dynamics of emigration of cells and then to fate restriction. But I am going to explain a little bit about potential and a little bit about fate. So in the lab, a student of mine, Shlomi Crispin, did the following. He labeled a cohort of cells or single cells. And he studied at the beginning, I don't want to say why. Basically, we were busy for more than 10 years in understanding the molecular network underlying emigration of cells, EMT, which appear to be very similar to cancer. A very similar network of molecules in the embryonic neural crest is responsible for generating movement of these cells. And after that, we became interested in understanding what is the dynamic of emigration, which types of cells emigrate when, and are they really multipotent or not. So what Shlomi demonstrated is that there is an inverse localization of cells in the tube that corresponds to their final location in the periphery. And let me show you this by the movie. Look at this. First, let me explain you that. The yellow cells are sympathetic progenitors. The green cells are Schwann cell progenitors. The blue cells are dorsal root ganglion progenitors. The red cells are melanocytes, or will be melanocytes. It's here. And the light blue cells will become the roof plate, which is the definitive dorsal portion of the tube after the crest cells have left. So nobody at this time knew what we found, that the roof plate originates basically ventral down to the cohort of crest cells, and as you will see here, will reach its dorsal position only after crest cells have left. Because people tend to think that the roof plate and the crest cells were one population, that everything was a mess in the dorsal neural tube, okay? And that cells leave as multipotent cells, and only after they colonize their ganglia, then they differentiate and they acquire an identity. And we found that this is not the case. So let me see how this maps. Look at this. The cells live like in a funnel type of mechanism. So I'll show it again. The yellow cells are the first ones to leave, and they will migrate. So yellow cells were the first. They will migrate the farthest and settle next to the dorsal aorta, okay, which is your very big ava urakin, your very big vessel, and then they will generate sympathetic cells. The next ones to leave will be the Schwann cells, then the RG, and then the melanocytes that change their migratory pathway. Instead of migrating down to generate neural cells, they migrate superficially under the skin to generate your melanocytes. So look at the order. I'll show it again once, and then we'll discuss it. Look at the order in which this happens. So there are two processes here. First, an ordered emigration of cells. It's not a random one. Cells that are going to be ventral were the most dorsal cells within the tube. Cells that were very ventral in the tube are going to be the most dorsal ones in the periphery. And there is a temporal order of colonization that matches the order of emigration from the tube. So there is no random thing here. There is quite a choice. And there is another thing. Look now at the tube, not at the periphery. Look at the tube. Sorry. Look at the tube. Once the cells leave, there is a ventral to dorsal relocation of progenitors. So it works like, think about a sand cloak, in which once the sand enters, okay, into the lower compartment, the other particles of sand will move towards it. So the cells do this and then they leave, like a funnel. So this was something which made a lot of noise in the literature. We found an inverse dorsoventral localization of progenitors in the tube vis-a-vis the derivatives. The delamination of the cells is continuous and is associated with the relocation from ventral to dorsal. Prospective roof plate cells, these ones, are localized ventral to the melanoblast, namely the definitive closure of the neural tube, when no more cells will leave it. The CNS, the spinal cord, will remain as such only after crest cells have left. Till that time, the roof plate is another lineage. And finally, based on this, and this is not what shows that, but I'll show you one example, we can predict the fate of a cell. The fate of a cell based on its localization in the dorsal tube and on its time of emigration. Okay.
Well, can you please go one click back just
Okay.
to see the
Right.
layers?
Sorry? Okay.
So what will happen, I don't know if it was tried or not, but what will happen if we'll take a population of neurons from, let's say, the red layer here, and implement that in the, let's say, green layer? Will they, in this stage, will they still change their fate, or how will they immigrate? Will they die? Was this studied?
Okay, so I will exemplify that in a minute, but unfortunately, technically, it's not possible in an epithelium to change a red cell for a blue cell. It's absolutely impossible, technically, because it's an epithelium. If you take a population, how do you know where do the red cells end and where do the blue cells begin? It's different. On the board, it's very easy.
Yeah, but I mean, I guess, so you can take them not from two nearby layers, but from too far away.
Yeah,
layers.
but you can't, because it's an epithelium. This is what I said about the clonal analysis in ovo. The epithelium is a tight epithelium. You know what is the size of this cell here? This is 5 microns. How do you take 10, 15 microns? We know to graft in a minimum of 50 microns. We can graft 50 microns, but 50 microns is a lot of cells. So you know what is this size? This is 100 microns in an embryo. 100 microns the entire tube. So it's technically impossible, and it is also an epithelium. But I'll show you what we did. Okay, what we did. So we approached this question obviously in another way, in a molecular way. כן, בבקשה.
Does it happen all simultaneously?
What do you mean?
Like you said, you can predict by localization.
Right.
But regarding to timing, what triggers it in
Okay, this is a beautiful question. The timing between this, the migration of the red cell and the migration of the yellow and the green is 2.5 days.
So once something triggers the yellow one, so 2.5 days later
it's a process. It's a gradual process. The first cells that you will see in the periphery are these ones, where the other ones are going still to be in the tube. And again and again, as time goes by, you are going to see, and we are talking about a particular level, right? Because at different levels there is a rostocodal gradient. So you have always to stick to a certain level of the tube. And then you can follow it with time. Because the embryo is a time machine. You will find all the different levels when you go from the tail to the head. But this is another complication I didn't talk about. Okay, but if you want to see this, you have to stick to a certain level and then follow it with time. So what we don't know, and I would like to know, and we can model it because we have the means, is how many of these progenitors do we have in the neural tube, because these cells proliferate. Throughout the time in which they live, they also proliferate. Very few of them die at this time, but they proliferate, and then they delaminate. Okay, how many of these progenitors do we have? How can we map the dynamic evolution of this system? These are very, very crucial questions. So let me just show you what we did to study fate, and then I'll show you and try to illustrate your question about potential. What will happen if we change the place of a cell? So how did we do this? Can you see this green dot here? Can you see that? So what Shlomi did is he decided to do a clonal analysis in vivo. How did he do it? Since we cannot graft a single cell, what he did is he injected a vector, a DNA, that encodes for green fluorescent protein, for GFP. And what he did then is to leave these cells, these embryos, to live for a certain amount of days. But the issue was to inject the dorsal tube here, only the midline, at 25 somites, namely when the embryo had 25 somite pairs at different ages, 27, 30, 35, and 40, only at the level of somite 23. So we are always dealing with a certain level. And what he found, which recapitulates what we saw in the movie, is that if he marks a single cell, look at this cell, this is 6 hours after when GFP is translated into protein. He finds that if he injects at 25 somites, mass city, if he injects that at 25 somites, all the cells he finds are in the sympathetic ganglion, namely here, next to the aorta. This is a magnification. But not in the roots, not in the DRG, and not as melanocytes. If he goes a little bit later, he finds them as Schwann cells. A little bit later, only in the DRG. A little bit later, only as melanocytes. And at the end, only as roof plate cells. The production of crest cells has ended, and the cells became only roof plates. So this means that the cells, and this was in more than 95% of his clones. So this was a work, this and a couple of other things, a work of 3 years, just doing this mindfulness. It's very difficult because when you do clonal analysis, you have one labeled cell in one of 10 embryos. Okay, the other embryos are going to be negative. So it's not easy at all doing this. The embryos are, the entire embryo is 1.5 millimeters. So you're working with a binocular. You're not working, it's not, this is not an embryo. Okay? So this means that the cells are fate restricted in terms of following the normal fate of a cell.
Sorry.
Okay.
Can you explain why?
What would be the alternative? What is your assumption when doing a clonal analysis? What are the possibilities?
What was the
Yeah, of
And,
course.
and every time he saw different expressions of.
What he does is he injects a label, okay?
Mm-hmm.
A label that is inherited by daughter cells.
Mm-hmm.
He injects it very early and at different time points, and he finds at different time points, let's say he has 10 embryos like this, 10 embryos like this, and he finds that if he injects very early, all the cells go to the sympathetic ganglion and not to the other derivatives. If he injects a little bit later, they go to the ventral root. Namely, they go to populate in a ventral to dorsal order. There is, again, order at the level of the single cell population. So this means that there is a ventral to dorsal order of colonization of cells because he injected at different times, and that the cells which leave the tube, the first ones go to the sympathetic, the last ones go to the melanocytes or to become roof plates. And what is most amazing to me is that one cell did not go to DRG and ventral root and melanocytes. They went at each time point to another different derivative, but coherent in their order. So this means that these cells are restricted. What this doesn't tell you is what? If this restriction is what?
From the beginning.
If it happens already, like if you can't see whether the cells by their makeup are already differentiated before migrating or only after.
Perfect.
Yeah.
So what you cannot know is whether restriction is imparted on the cell by the environment that basically changes from this stage to this stage, because you can argue, no, the cell is multipotent, but because the environment changes, and believe me, it changes, the cell is driven to go to another place. So at the beginning, there is a highway to the sympathetic. At the end, everything is busy, and you can go to the melanocytes only. Or whether the makeup of the cell is different, namely intrinsic versus extrinsic environmental conditions. So what this experiment says, perfect. What this experiment says is that there is phase restriction, but it doesn't tell us whether it is intrinsic or extrinsic. And this is where we go to study potential. So what did we do? What one can do in this type of cases, and this is, with this I am going to end my talk. We used the following paradigm. We basically took advantage of the fact that crystals that give rise to neural derivatives migrate ventrally through like 5 different migratory pathways. It doesn't matter now. But cells that live one day later, when these are already populated, will go through the ectoderm, under the ectoderm, namely laterally. So they will change their pathway from here to there. And the melanocytes obviously have to end up in the skin, so they migrate superficially. So what we did is we took neural cells, we transfected them by electroporation. We can make, by virtue of a certain current that we give the cells, okay, we call it the electric chair, it's called electroporation. We can basically deliver different genes. So what we did is we gave the neural progenitors a gene that encodes either for GFP, this is the normal control, or for a gene that is called endothelin receptor B with GFP. We cloned it fused in frame to GFP. So EDNRB is known in development to guide the cells to migrate laterally, namely to the melanocytic pathway. It has only a guidance function. So we ask, if we divert the migratory pathway of the cells from a ventral to a lateral location, will the neural cells, or the cells which would otherwise become neural, will they become melanocytes? This is exactly what you ask. Now we perform the electroporation in the tube, so we know exactly where we work on. We cannot change their place, but we can mark them differentially. Okay, so this will again hopefully answer your question. So look at the results. These are the normal cells. They migrate ventrally. These are are cells that have been diverted into the lateral pathway. You see them with the arrowhead. This one and this one and these ones here. They express EDNRB2, and they are also green. So we ask the question, and here you can see that like 35% of the cells can be diverted to become ectodermally migratory cells, or subectodermally. And then we ask, are they melanocytes or are they neurons? Okay? Of course, some of them will still be here, never mind. So if we look at MC1, which is a melanocyte marker, we notice that these cells are MC1 negative, MELM negative, and c-KIT negative. These three markers are melanocytic markers. So even if we drive these cells to go to the subectodermal pathway, they are not going to become pigments. Do they upregulate de novo neural markers? The answer is yes. So what you see here, just look at this panel. These cells that migrate superficially express CASH1 and HNK1. This is CASH1. It's a proneural transcription factor, one of the earliest, that characterizes the sympathoadrenal lineage. They express islet-1, which you can see also here in a dorsal root ganglion. They express FOXD3, which is a very important transcription factor, expressed only in neural progenitors. You see this, you see FOX. They express TRAC-C, the neurotrophin receptor. You can see it here in these cells migrating under the skin. And they express a fully differentiated marker of neurons, which is neurofilament. You can see neurofilament in these cells. This means that even after changing their environment, these cells still upregulated neural markers, namely they stuck to their original identity. Now we said, maybe we brought them into the melanocytic pathway too early. So let us bring them later, when the environment of the embryo is busy with making melanocytes, because melanogenesis is a late process. So what we did is we combined electroporation with grafting. And we did the following. We took a very early donor, a quail donor, and we transfected it with either GFP or EDNRB2. And then we grafted it into a very old host. You see, the host is very old. It's busy with melanogenesis. And the donor is very young, only 18 somites. And as you can see, we put the tube. You can see the hole is much bigger because the embryo is much bigger. But after 20 hours, everything regenerates. The embryo regenerates beautifully. So I would like to show you what happens under normal conditions if we do not perform EDNRB. The red cells of the quail, now recognized by the QCP and antibody, the red cells of the quail, look at this beautiful graph. Look at the size of the DRG of the host. It's like the size of the tube of the donor, because the host was enormous. So this, what you see is that these cells, the red cells, never went to the melanocyte pathway. They went directly to the DRG of the host, where some of them even differentiated as neurons and expressed markers. So then we said, okay, now we are going to transfect or to graft tubes that were transfected with EDNRB. Namely, we are going to oblige the cells to go to the melanocytic pathway. Are they going to become neurons or melanocytes? To make a long story short, they become neurons. So even if, and you can see it here, these are cells going into the dermis prior to the epidermis. They express FOX, they express neurofilament, these ones, they express also CASH1, and they express islet. So this means that even after forcing the cells, no matter what the time is, even under the best condition of melanogenesis, neural progenitors are phase restricted because of intrinsic reasons, not because the environment dictates their fate. Now, having said that, I'm not going to tell you that, of course. We have found the factors that are responsible for that. We have found the network of factors, for example, FOXD3, SOX9, SNAIL2. These this trio of factors is expressed in the dorsal tube. In the dorsal tube, in all the progenitors, melanocytes and also neural progenitors. But when the neural progenitors left the tube and only the melanocytes are still there, fox, sox, and snail disappear. They are completely downregulated. So there is a molecular difference between neural and melanocyte progenitors already in the dorsal neural tube prior to emigration. So this restriction of fate is cell intrinsic. It does not depend on the initial migratory pathway and is identified at least by 3 different transcription factors. So now I'm going just to show you how we demonstrate using the mouse embryo and genetic means that FOXD3 that we found to act downstream of the other 2, I'm not going to bother you with this, how this molecule may change the fate of cells. Because if we give FOXD3 to the prospective melanocytes, they will transform into glia. And if we create an embryo that is a mutant to FOXD3, this is best done in the mouse, okay? It's never mind the genotyping here. It is a mutant mice that we constructed in which FOXD3, which is a very important transcription factor in the embryo, is mutated only in the crest, not in other cells of the body. So what would be your prediction? If FOXD3 inhibits melanogenesis and promotes neurogenesis, what happens if we delete FOXD3?
The mouse will have no pigment cells.
On the contrary. On the contrary, FOXD3 is expressed in neural progenitors, never expressed in melanocyte progenitors. If we give FOXD3 to prospective melanocytes, we get glia instead of melanocytes in the chick embryo. What will happen if we delete FOXD3 at all from the crest? If FOXD3 is an inhibitor of melanogenesis,
You get melanocytes instead of
in neurons. So this is exactly what we got. So what we get is the following, and I'm showing you just one picture. This is a normal control mouse. You can see in green, this is YFP. You can see melanocytes that begin leaving. In blue, this is MITF. MITF is the master generator of melanocytes. It's called microphthalmia gene. And you see the melanocytes, how they begin leaving the dorsal tube, but you can never see melanogenesis in the dorsal tube. The dorsal tube is made only of progenitors. Whereas now in the mutants, we begin seeing melanogenesis, pigment cells, within the neural primordium. This is never ever seen in a normal embryo. And then if we look at an older embryo, this is a control, you see melanocytes under the skin, and you see a normal dorsal root ganglion, right? And this color means TUJ1. TUJ1 is a tubulin. It's a neuronal marker. And look what you see in a DRG of the mutant mice. You see a lot of melanogenesis at the expense of both glia and neurons. You see a very, very much enhanced melanogenic process at the expense of glia. And this means, to make the summary of all that I told you, that in vivo, contrary to in vitro, the cells basically find a certain environment that they follow. We believe that for neural crest cells in the trunk, the dorsal neural tube is the first environment that acts somehow in an instructive manner to help the cells determine what they will become. Maybe these are gradients of BMP, which is the molecule that is localized in the dorsal tube. We are trying to understand that. Modeling would be fantastic here, because pretty much the same as sonic is graded in the ventral tube up to the dorsal, BMP is a gradient from the dorsal to the ventral. But based on the dynamic behavior of the crest, which leaves the tube, you would argue, this is so far for the data, now I am hypothesizing. You would argue that if there is a gradient of BMP, very high here and lower here, that the yellow cell would get exposed to the highest concentration of BMP, but for the shortest time, because it's the first cell to leave. And this cell here would be exposed to the lowest amount of BMP, but for the longest time, because this cell will still have to go במעלה gradient, okay, upstream to the gradient of BMP before leaving the dorsal tube. So how can I model, how can I understand mathematically which cell will get what and whether these cells will change their behavior? How can I don't know how to do that, because one has to take into account the amount of BMP which exists there, the time that is spent by a certain cell in a certain place in the dorsal neural tube, namely the rate at which the cell leaves the neural primordium, the rate at which it proliferates, and we know that because we have studies of BRDU labeling and so on. So there is a lot to be done here to try and understand many, many important events leading to the formation of the peripheral nervous system and the final separation of the peripheral nervous system from the central nervous system, because the roof plate, which is left after the exit of peripheral cells, this is the final closure of the CNS. And once you have this roof plate becomes a very crucial central organizer, because the roof plate is going to continue secreting BMP, and it is going to act upon the lateral cells here and to create all the interneurons that you have in the CNS. DI1, DI2, DI3, 4, 5, and 6. All the interneuronal types that you have are induced by this structure, which is the roof plate. So one of the main projects in my lab is now trying to understand not the beginning of the lamination of crest cells, which we have worked on for many years, but what triggers the end of the process of crest production and the transition of the dorsal neural tube from PNS to CNS. Okay? Any questions?
So we just saw that at least we can know for when the neuron, the progenitors or neurons, or I don't really
Progenitors.
know, the progenitors are leaving the place, so they are already determined.
I would say determined. I would say restricted, because we really do not, for example, when we map a single cell in the dorsal tube and they go to the DRG, they will still give rise in the DRG to neurons and glia. So they are not fully determined to give rise to a TRAC-C positive neuron in the ganglion. No, they are restricted. They are biased to go to the DRG, to become sensory. But within the sensory lineage, they can still become a neuron of different types and the glial. So I prefer the word restricted.
Okay. My question is, as far as you know, is that the same as adult neurogenesis? Or
This is a very